Hello everyone, my name is Mollie Wilson and I'm a final year PhD student in Luke Bilter's lab at the University of Edinburgh. And in Luke's lab, we're mostly focused on looking into disease and development of the liver. And I was fortunate enough to be on a funded studentship with the AMMF, so I'm looking at intrahepatic cholangiocarcinoma. And I'm just delighted to be able to update you on what I've been doing for the last 3 years. So I'm going to talk today about my project, which is identifying tumour suppressors that interact with oncogenic Kras G12D in intrahepatic cholangiocarcinoma. So as we all know, intrahepatic cholangiocarcinoma, or ICC, is a lethal malignancy of the peripheral bile ducts, and it presents with these highly genetically heterogeneous tumours, of which there are some kind of canonical drivers, which I've sort of listed here. But these are of a limited number and they're relatively inconsistent between patients. And on top of this, as the rarity of this disease means there's a hugely limited amount of tumour tissue available for genetic analysis, there really does present a problem that ends up impeding treatment. So to address this kind of gap in the understanding of the functional genetics of ICC, we decided to pool the limited amount of exome sequencing data that we have, which is from 5 different published studies, and to basically collate and reanalyze this. So this is work done by an ex-PhD student in my lab, Nicholas Younger, and they actually presented here at AMMF in 2019. So I'm just going to go over their work briefly. Yeah, basically, so we had this exome sequencing data from these 5 different studies, and Nick designed this computational pipeline where we could, collate and reanalyze this data. So this led to the rediscovery of some canonical drivers of ICC, so including, for example, gain-of-function mutations in the IDHs and in Kras, and loss-of-function mutations in ARID1A and TP53. So the screen also drew out these loss-of-function mutations that are already present in the COSMIC database, so the catalogue of somatic mutations in cancer. And most interestingly, there are also around 90 novel driver genes. So of these 90 loss-of-function mutations, it really demonstrates how ICC in humans is typified by a large number of infrequently mutated genes. So that's what these are here. All these are genes along the bottom. And some of these genes are only present, gene mutations are only present in 1 to 2 patients. So I also want to bring to your attention that even the most highly mutated genes, so oncogenic mutations in Kras and loss of function in TP53, these are still only present in around 17% of patients, which is interesting to us because we know that Kras and TP53 alone are sufficient to form ICC in vivo. So we're wondering, what are these other mutations doing if these alone are enough? So we also know that upon aberrant oncogenic aberrant oncogene activation in the liver, so for example in Kras, cells enter this state called senescence surveillance. So this is a protective mechanism against tumorigenesis, where the senescing cells will then produce cytokines and growth factors, and it targets them for immune clearance and apoptosis. So Kras cells are cleared from the liver by this mechanism, and we know that the additional loss of TP53 means cells are able to circumvent this protective mechanism, and that means that they're then retained in the liver, and then they can proliferate and become tumorigenic. So this really leads into my project, where we can then ask, are any of these infrequently mutated genes that came out of the computational screen able to allow a cell that's already expressing oncogenic Kras to then become proliferative and initiate ICC in a similar way that TP53 does? So are these mutations capable of enhancing or modifying ICC growth? So to address these questions, I decided to validate Nick's screen in vivo using our murine model. So to do this, I used a CRISPR-Cas9 library. So I cloned these 90 novel gene mutations into I've cloned the 90 loss of function genes that we identified with mutations in the computational screen into this Sleeping Beauty CRISPR system, where there's a guide scaffold that will allow the stable integration into the genome of the guide RNA, and that will result in CRISPR targeted Cas9 editing when we co-inject these plasmids with Sleeping Beauty 13 transposon. So we additionally used a Kras GFP plasmid that contains this internal ribosome entry site. That means we can have, I can basically visualize the Kras positive cells because they'll be expressing GFP. So I have my library of these guide RNAs against the genes identified in the computational screen, and I have the oncogenic Kras, and we injected these into the tail veins of mice using this hydrodynamic tail vein injection. And this system means that you can introduce these naked DNA plasmids in a way that you do it in such a large volume that it kind of overburdens the cardiovascular system of the mouse. And this means that the liver becomes more leaky, if you like. So the hepatocytes, they will uptake the naked DNA, and it means that you will have these plasmids expressed in the liver. So 8 weeks post-injection, we sacrificed the animals and found that the animals injected with Kras alone had no tumours, but those with the library and Kras had macroscopic tumours. And this is just a little immunohistochemistry here for GFP to show that Kras is expressed. And these tumours were actually sort of highly representative of human ICC in that they were highly stromatic, and they were surrounding these kind of ductular and granular structures. So we then did some exome sequencing, firstly just to confirm that the indels in these tumours were indeed caused by induced by CRISPR-Cas9 editing. So we just did this by showing that they are within 50 base pairs of the PAM site. And we also did exome sequencing just to see what mutations were present. And we found that in all of these tumours, there were loss of function mutations in TP53. So this made us want to establish a model of ICC with oncogenic Kras and TP53 that would act as our kind of background cancer model for this work, if you like. So to do this, I just did a clone. I just used a single guide RNA against TP53 and the same oncogenic Kras as before, and injected these hydrodynamically into mice with Sleeping Beauty 13 transposon as before. So establishing this model gave us a great control to use when we did RNA sequencing to look at differential expression of transcripts in our CRISPR screen tumours compared to this Kras TP53 control. So I also did RNA sequencing compared to normal bile ducts that we isolated from these immune compromised mice that we use for this experiment. But we figured that because all of our screen tumours contained already Kras and loss of function in TP53, that would be a better control to use. And so we're interested on what mutations occurred on top of this phenotype. So anyway, we found this, and there was a subtype of tumours that clustered tightly, significantly on principal component analysis. And we found that the common mutation between them is in this gene called neurofibromin 2. So neurofibromin 2 encodes for a protein called Merlin, which is a cytoskeletal protein. It's part of the firm domain protein family. And neurofibromin 2 is most associated with its namesake disease, neurofibromatosis 2, which is the development of benign tumours of the nervous system, which usually affects Schwann cells. So most interestingly, its encoded protein Merlin is at the nexus of many signaling pathways, just as you can see here. and its dysregulation has been observed in many other cancer types. So next I wanted to see whether NF2 loss would enhance or modify our Kras T53 model. So again, using our hydrodynamic tail vein injection model, I found that the additional loss of NF2 on the background of my Kras, oncogenic Kras, and loss of TP53 significantly accelerates mortality in the animals, shown by the blue here on the survival curve. So, these animals were coming down in as little as 11 days compared to the Kras TP53 control, which came down, sort of around 40 days. Additionally, I found that when I targeted NF2 for CRISPR-mediated editing alone, these animals also developed tumours, even in the absence of TP53. So that's here. There's just some H&E stains to give an idea of what the tumours look like. So the tumour number in the dual loss mice was greatly higher, but the overall area of the mouse covered by tumour was adjusted as a measure of a percentage tumour of the whole area of the liver, if you like, was still greatest in the control mice, so the Kras TP53. And we also included a scrambled guide control, which is shown in grey here. So I then wanted to do some protein and RNA transcript analysis on these to compare the different models and look into the phenotypes. So first of all, RNA sequencing analysis allowed me to look at the differential expression of transcript levels. So we found that Wnt and cadherin signaling components were downregulated in animals with loss of NF2. So canonical Wnt beta-catenin signaling is aberrant in the developing liver, particularly during embryogenesis, and then mostly it's inactive in the mature healthy liver, apart from its sort of limited roles in regulation of liver homeostasis and repair. So it's highly conserved and is a very well-controlled pathway, and as such it has great importance. So abnormal Wnt beta-catenin signaling has been shown to drive cholangiocarcinoma development. Yes. So we also can see here that there's a great number of protocadherins that were downregulated, and protocadherins have been shown to regulate Wnt signaling as well, and this is by their way to act as tumour suppressors through inactivation of the tumorigenic Wnt signaling pathway. Yes. So, and also we have upregulation of Wnt9b, which is an activator of canonical Wnt beta-catenin signaling. So beta-catenin is the core protein involved in the Wnt beta-catenin signaling pathway, and in the absence of Wnt ligands and the canonical Wnt pathway is off, beta-catenin is phosphorylated and it's targeted for destruction. So and we also have these negative regulators of Wnt signaling here that are downregulated, so NKD2 and SFRP1, for example. These data were further supported when I did some proteomic analysis of the tumours from these three models as well. And I did this using a reverse phase protein array, which is basically you extract protein from all of your tumour samples and attach these to a chip and then wash them with chosen antibodies. So I chose antibodies associated with all the different signaling pathways that I was interested in. So the proteomic analysis supported the RNA-seq in the sense that there was upregulation of this glycogen synthase kinase 3 beta, which is a component of the beta-catenin destruction complex. And this is really important for the Wnt beta-catenin pathway, maintaining an off state, because when there's no Wnt ligands present, dysregulation of the beta-catenin destruction complex is showing basically that tumours with NF2 loss are having this more active Wnt state. So likewise, beta-catenin itself is also dephosphorylated, so therefore in its active state, and you can see that it's downregulated also in tumours with loss of NF2. And finally, the other component we found very interesting in the RPPA analysis was the stark upregulation of this active phosphorylated AKT, which is indicative of increased PI3 kinase AKT signaling, and this is an essential pathway for apoptosis to occur. So this provided us with enough evidence to look back at our patient data, published patient data, and we looked back at this patient transcriptomic data, and particularly we're interested in the Wnt AKT status of patients' tumours. We identified that there's this subgroup of patients that have increased coactivation of Wnt and AKT signaling, and based on this, and combined with our in vivo studies, we believe that these patients might deal from a dual therapeutic approach. So we decided to test this in our mouse models, and here I've just shown a rough working model on the mechanism that I propose may be occurring so far. So you can see that all these components that are in bold are the ones that we have evidence are that we see differential changes in either their protein or transcript levels. So essentially, we end up with these two main outcomes. So either the stabilization of beta-catenin, which means that when it's not destroyed, you end up with its migration to the nucleus, and that allows increased Wnt signaling and therefore increased tumorigenesis. Or you end up with suppression of apoptosis, which we believe is done through this path, this arm here of increased AKT signaling that phosphorylates FOXO-As, which then deactivate apoptosis. And it means that you end up with suppression of apoptosis. And to confirm whether this is a realistic model in a therapeutic context, we treated our aggressor model with a porcupine inhibitor, LGK974, and a PI3 kinase inhibitor, which is a key component of the AKT signaling pathway. So I believe porcupine inhibitors are currently in clinical trials, and PI3 kinase has been improved for other cancer types, but not yet been used in ICC. So you can see here that the combination treatment in red, the mice treated with both drugs had a significantly longer mortality than those with either drug individually or with the vehicle. So the final thing that I did was to test this in another ICC model, which was more true to human disease in that it arises on the background of chronic inflammation. So this model is quite clever. It allows CRE to be targeted to K19 expressing cells, so to the bile duct, and it allows the excision of an exon from P10 and from P53, which knocks them out. And it also knocks out a stop codon for a tomato expressing gene, meaning that the cancerous cells are tomato positive, as you can see here, and can be visualized very easily. So we just did some staining as well to show that in this different model, you still have overactivation of beta-catenin, dephosphorylated beta-catenin is active, and phospho-AKT, which is also active. So yeah, and then we treated these with the porcupine inhibitor and the PI3 kinase inhibitor as before, and you can see that the inhibition of Wnt and PI3 kinase signaling substantially reduces tumor growth, and indeed 2/3 of the animals treated with these drugs were completely tumor-free, compared to 1/5 of the control. So in summary, I used CRISPR-Cas9 screening of these loss-of-function mutations that we identified with the computational screen to identify NF2 as a rare driver of ICC. And we found that deletion of NF2 interacts with oncogenic Kras G12D to initiate ICC in vivo, and that NF2 also interacts cooperates further with TRP53 loss, and that leads to this greatly accelerated phenotype with high tumor genesis and increased mortality. And then the proteomic and RNA sequence analysis showed that coactivation of Wnt and PI3 kinase signaling occur, and that these pathways can both be targeted in vivo, and it greatly extends mortality into animal models. So I'm just going to very quickly thank the AMMF for funding all of this great work, and thank you to my lab and all of the other people who helped contribute to this project. Thank you very much.